ChatGPT said. Histochemical and immunotechniques. Histochemical and immunotechniques are essential tools in biological research, pathology, and diagnostic medicine. They allow visualization, localization, and identification of specific molecules, structures, and cellular components using specialized staining and antibody-based detection methods. HISTOCHEMICAL techniques. Histochemistry is the study of the chemical composition of cells and tissues using specific staining techniques. These methods help in the identification of enzymes, carbohydrates, lipids, proteins, and other cellular components. 1. Principles of histochemistry. Histochemical techniques rely on the chemical reaction between cellular components and a staining reagent, the formation of an insoluble, visible product at the site of reaction, the specificity of staining for particular biomolecules. 2. Common histochemical stains and their applications. Hematoxylin and eosin H&E staining. Hematoxylin stains nuclei blue, binds to nucleic acids. Eosin stains cytoplasm and extracellular proteins pink. Used for general tissue morphology and histopathology. Periodic acid shift staining. Stains carbohydrates and mucopolysaccharides magenta. Used to detect glycogen, basement membranes, and fungal infections. Sudan black and oil red O staining. Stains lipids in cells and tissues. Used in fat metabolism studies and lipid storage disorders. Congo red staining. Identifies amyloid deposits in tissues red under normal light, green birefringence under polarized light. Silver stains reticulin, gomorrhes, methenamine silver. Used for detecting reticular fibers, fungi, and certain bacteria, e.g., Trichomema pallidum. Prussian blue reaction, pearl stain. Detects iron deposits in tissues, used in the study of iron metabolism disorders. Fulgen stain. Stains DNA specifically red, used in nuclear studies and cell cycle analysis. Alcyon blue stain. Detects acidic mucopolysaccharides and glycosaminoglycans in tissues. 3. Enzyme histochemistry. Enzyme histochemistry uses enzymatic reactions to visualize cellular structures based on enzyme activity. Common examples include alkaline phosphatase ALP staining, detects osteoblast activity in bone tissue. Acid phosphatase staining, identifies lysosomes and autophagic vesicles. Myeloperoxidase MPO staining, used for detecting granulocytes in hematology. 2. IMMUNOTDCHNIQUES. Immunotechniques use antigen-antibody interactions to detect and localize proteins, pathogens, and specific cellular markers. These methods are crucial in diagnostics, cell biology, and molecular research. 1. Principles of immunotechniques. Antibodies recognize and bind to specific antigens, proteins, polysaccharides, or other biomolecules. Specificity of antigen-antibody interaction allows precise detection and localization. Visualization is achieved using fluorescent dyes, enzyme-linked reactions, or radioactive labels. 2. Types of immunotechniques. A. Immunohistochemistry IHC uses antibodies to detect proteins in tissue sections. Enzyme-linked or fluorescent detection methods highlight antigen-antibody interactions. Applications. Cancer diagnosis e.g., HER2 in breast cancer. Neuropathology e.g., tau and beta amyloid in Alzheimer's. Infectious disease detection e.g., viral proteins in tissue. B. Immunocytochemistry ICC. Similar to IHC but performed on isolated cells instead of tissue sections. Used for analyzing protein expression in cultured cells or cytological samples. C. Enzyme-linked immunosorbent assay ELISA quantitative technique for detecting proteins, hormones, and antibodies in fluids. Types. Direct ELISA detects antigen using a labeled primary antibody. Indirect ELISA uses an labeled primary antibody and a labeled secondary antibody. Sandwich ELISA uses two antibodies to capture and detect an antigen. Competitive ELISA measures antigen concentration by competition with a labeled antigen. Applications. Diagnosis of infections e.g., HIV, COVID-19. Hormone level assessment e.g., insulin, thyroid hormones. D. Western blotting. Detects and quantifies specific proteins in a mixture. Steps. Protein separation by SDS phage. Transfer to a membrane nitrocellulose PBDF. Incubation with primary and secondary antibodies. Detection by chemiluminescence or fluorescence. Applications. Detection of viral proteins e.g., HIV confirmation test. Analysis of protein expression in research. E. Immunoprecipitation IP and company immunoprecipitation company IP used to isolate specific proteins or protein complexes from cell lysates. Applications. Studying protein-protein interactions. Identifying signaling pathway components. F. Flow cytometry. Uses fluorescent labeled antibodies to analyze cells in suspension. Allows measurement of cell size, granularity, and surface intracellular markers. Applications. Immunophenotyping in leukemia, lymphoma. Analysis of apoptosis and cell cycle. G. Immunofluorescence microscopy IFM uses fluorescently labeled antibodies to detect proteins in cells or tissues. Types. Direct immunofluorescence DIF labeled primary antibody binds directly to antigen. Indirect immunofluorescence IIF in labeled primary antibody is followed by fluorescent secondary antibody. Applications. Autoimmune disease diagnosis e.g., detection of antinuclear antibodies in lupus. Subcellular localization of proteins. 3. Comparison of histochemistry and IMMUNOTDCHNIQUES feature histochemistry immunotechniques. Principal chemical reactions with cellular components antigen-antibody interaction specificity moderate depends on reaction conditions high antibody-based visualization colorimetric stains fluorescence enzyme-linked chemiluminescence applications tissue morphology enzyme localization protein detection disease diagnosis molecular research IV applications in research and medicine medical diagnostics histochemistry is used in pathology labs for disease diagnosis immunotechniques like ELISA and Western blot are widely used for infectious disease detection cancer research IHC helps in identifying cancer markers e.g. P53 Ki67 flow cytometry is used for leukemia classification neuroscience immunofluorescence detects neurotransmitters and brain proteins histochem Histochemical stains identify neurodegenerative disease markers. Infectious diseases. ELISA and Western blot detect viral and bacterial infections. IHC identifies microbial antigens in tissue samples. Genetic and developmental studies. Immunotest track protein expression during development. Histochemical stains analyze metabolic processes in embryos. B conclusion. Histochemical and immunotechniques are powerful tools in biological research and diagnostics. Histochemistry provides insights into cellular composition, while immunotechniques enable specific detection of proteins and antigens. These methods have revolutionized pathology, molecular biology, and clinical diagnostics, contributing to advances in medical science and research. You said, antibody generation. ChatGPT said, antibody generation. Antibody generation is the process of producing antibodies that specifically recognize and bind to antigens. Antibodies, immunoglobulins, Ig, are essential components of the immune system and are widely used in research, diagnostics, and therapeutics. Immunological basis of antibody generation. Antibodies are produced by B lymphocytes, B cells, in response to foreign molecules, antigens. The process involves antigen recognition. B cells recognize antigens through membrane-bound immunoglobulins, B cell receptors, BCRs. B cell activation. Activation occurs via T cell-dependent protein antigens or T cell-independent polysaccharides, lipids mechanisms. Helper T cells, CD4, release cytokines to stimulate B cells. Clonal expansion and differentiation. Activated B cells proliferate and differentiate into plasma cells, antibody-secreting cells, and memory B cells, long-term immunity. Antibody production. Plasma cells produce large quantities of antibodies specific to the antigen. The initial antibody response is IgM, followed by class switching to IG, IgA, or IgG. 2. Methods of antibody generation. 1. Polyclonal antibody production. Polyclonal antibodies, PABs, are a mixture of antibodies targeting multiple epitopes of an antigen. They are generated in animals such as rabbits, goats, sheep, or mice. Steps. Antigen preparation. Purified protein, peptide, or whole cell lysate is used as the antigen. Immunization. The animal is injected with the antigen along with an adjuvant, e.g., Freund's adjuvant, to enhance the immune response. Multiple booster injections are given at regular intervals. Blood collection and serum preparation. Blood is drawn after an immune response has developed. The serum, containing antibodies, is collected and purified. Purification of antibodies. Affinity chromatography, protein AG, antigen-specific columns, is used to purify antibodies. Advantages. Recognizes multiple epitopes, right pointing arrow, higher sensitivity. Faster production, few weeks. Useful for detecting denatured proteins. Disadvantages. Higher batch-to-batch variability. May contain non-specific antibodies. 2. Monoclonal antibody, MAB production. Monoclonal antibodies recognize a single epitope on an antigen and are produced using hybridoma technology, Kohler and Milstein, 1975. Steps. Immunization. A mouse is injected with the antigen multiple times to stimulate B cell production. B cell isolation. Spleen cells rich in B cells are harvested. Fusion with myeloma cells. Spleen B cells are fused with immortal myeloma cells using polyethylene glycol PEG. The resulting hybridoma cells can continuously produce antibodies. Selection and screening. Hybridomas are selected using HAT medium, hypoxanthine ammonopterin affinity, which only allows fused cells to survive. Cells producing the desired antibody are screened using ELISA or Western blot. Cloning and expansion. Positive hybridoma clones are expanded in culture. Large-scale production is done in bioreactors or acidic fluid in mice. Purification. Antibodies are purified using affinity chromatography. Advantages. High specificity, binds only one epitope. High reproducibility, consistent batch quality. Can be engineered for therapeutic applications. Disadvantages. Expensive and time-consuming, approximately 6 months. Requires specialized hybridoma facilities. 3. Recombinant antibody production. Recombinant antibodies are generated using molecular cloning and expression in host cells, e.g., bacteria, yeast, or mammalian cells. Steps. Antibody gene cloning. Antibody genes, variable regions of heavy and light chains, are cloned into an expression vector. Expression in host cells. The vector is introduced into E. coli, yeast, or mammalian cells for large-scale production. Purification. Recombinant antibodies are purified using chromatography methods. Advantages. High specificity and reproducibility. Can be humanized for therapeutic use. Scalable for industrial applications. Disadvantages. Requires genetic engineering expertise. Expensive production. 3. Types of engineered antibodies. Humanized monoclonal antibodies. Mouse antibodies are modified to resemble human antibodies, e.g., trastuzumab for breast cancer. Chimeric antibodies. Contain a combination of mouse and human antibody regions, e.g., rituximab for lymphoma. Single-chain variable fragment, SCFV. Small antibody fragments useful for targeted therapy. Nanobodies. Derived from camelid antibodies, stable and highly specific. IV. Applications of antibodies, medical diagnostics. ELISA, Western blot, and IHC for disease detection. COVID-19, HIV, and cancer biomarker detection. Therapeutics. Monoclonal antibodies for cancer, e.g., pembrolizumab, bevacizumab. Autoimmune disease treatment, e.g., infliximab for rheumatoid arthritis. Research applications. Flow cytometry, immunoprecipitation, and immunofluorescence. Vaccine development. Antibodies are used in passive immunization strategies. B conclusion. Antibody generation is a crucial process in immunology, biotechnology, and medicine. While polyclonal antibodies offer broad specificity, monoclonal and recombinant antibodies provide high specificity and reproducibility. Advances in antibody engineering continue to revolutionize diagnostics and therapeutics, making them invaluable tools in modern science. You said. Detection of molecules using ELISA. ChatGPT said. Detection of molecules using ELISA. Enzyme-linked immunosorbent assay, ELISA, is a widely used biochemical technique for detecting and quantifying molecules such as proteins, peptides, hormones, antibodies, and antigens. It is highly sensitive, specific, and adaptable for clinical diagnostics, biomedical research, and pharmaceutical applications. I principle of ELISA. ELISA is based on antigen-antibody interactions coupled with an enzymatic reaction that generates a detectable signal, usually colorimetric or chemiluminescent. The key steps include antigen or antibody immobilization on a solid surface, usually a 96-well polystyrene plate. Binding of a specific antibody to the target molecule. Enzyme conjugation, enzyme-linked secondary antibody. Substrate addition, leading to a color change, detectable via spectrophotometry. The intensity of the color change is directly proportional to the amount of the target molecule present. 2. Types of ELISA. 1. Direct ELISA. Principle. The target molecule antigen is directly bound to the plate, and a single enzyme-linked antibody binds to it for detection. Steps. Antigen is coated onto the microplate. Enzyme conjugated primary antibody is added. Substrate is added, and color develops. Advantages. Simple and fast. Disadvantages. Lower sensitivity and specificity, higher background noise. 2. Indirect ELISA. Principle. Uses an labeled primary antibody to bind the target molecule, followed by an enzyme-linked secondary antibody. Steps. Antigen is coated onto the plate. Primary antibody binds to the antigen. Enzyme-linked secondary antibody binds to the primary antibody. Substrate is added for color development. Advantages. Higher sensitivity, amplification via secondary antibody. Disadvantages. More steps, longer procedure. 3. Sandwich ELISA. Principle. Uses two antibodies, capture and detection antibodies, to detect the target antigen. Steps. Capture antibody is coated onto the plate. Antigen is added and binds to the capture antibody. Enzyme conjugated detection antibody binds to the antigen. Substrate is added for color development. Advantages. High specificity and sensitivity, used for low concentration proteins. Disadvantages. Requires two antibodies, making it more expensive. 4. Competitive ELISA. Principle. Measures antigen concentration by competition between free and bound antigen for antibody binding sites. Steps. Antigen is pre-incubated with a known amount of enzyme-labeled antibody. The mixture is added to an antigen-coated plate. The more antigen present in the sample, the less antibody binds to the plate. Substrate is added, and the signal is inversely proportional to antigen concentration. Advantages. Suitable for small molecules and hormones. 4. Competitive ELISA. Principle. Measures antigen concentration by competition between free and bound antigen for antibody binding sites. Steps. Antigen is pre-incubated with a known amount of enzyme-labeled antibody. The mixture is added to an antigen-coated plate. The more antigen present in the sample, the less antibody binds to the plate. Substrate is added, and the signal is inversely proportional to antigen concentration. Advantages. Suitable for small molecules and hormones. Disadvantages. Requires precise standardization. 3. Detection systems in ELISA colorimetric detection. Uses chromogenic substrates, e.g., TMB or OPD, that change color upon enzyme action. Absorbance is measured using a spectrophotometer. Fluorescent ELISA. Uses fluorophore-labeled antibodies. Higher sensitivity than colorimetric ELISA. Chemiluminescent ELISA. Uses chemiluminescent substrates, e.g., luminol that emit light upon enzymatic reaction. Highly sensitive, used in clinical diagnostics. IV. Applications of ELISA medical diagnostics. Detection of infectious diseases, HIV, hepatitis, dengue, COVID-19. Measurement of hormones, insulin, HCG for pregnancy tests. Identification of autoimmune disorders, rheumatoid factor detection. Biomedical research. Quantification of cytokines and growth factors. Study of antibody-antigen interactions. Food and environmental testing. Detection of food allergens, gluten, peanut proteins. Testing for contaminants, pesticides, toxins. Drug development and pharmacokinetics. Monitoring therapeutic drug levels in patients. Screening for drug antibodies in biologic therapies. The advantages and limitations of ELISA. Advantages, limitations. High sensitivity and specificity requires specialized equipment. Plate reader quantitative and qualitative analysis. Cross-reactivity may lead to false positives. Scalable for high throughput. Screening time consuming. Multiple incubation steps. Relatively low cost compared to other techniques. Requires optimization for best performance. Conclusion. ELISA is a powerful tool for detecting and quantifying biomolecules with high specificity and sensitivity. Its versatility makes it indispensable in clinical, research, and industrial applications. By selecting the appropriate ELISA type and detection method, researchers and clinicians can obtain reliable and reproducible results for various biological and diagnostic assays. You said, RIA. ChatGPT said, radioimmunoassay, RIA. Radioimmunoassay, RIA, is a highly sensitive technique used to detect and quantify minute concentrations of biomolecules, such as hormones, proteins, and drugs, using radioactively labeled substances. Developed by Rosalind Yalow and Solomon Burson in the 1950s, RIA is based on antigen-antibody interactions and the competitive binding principle. Principle of RIA. RIA works on the principle of competitive binding, where a radioactively labeled antigen tracer competes with an labeled antigen sample for binding to a specific antibody. The amount of radioactivity measured is inversely proportional to the concentration of the labeled antigen in the sample. Key components. Antigen analyte, the molecule to be detected, e.g., insulin, thyroxine. Radioactive tracer, the same antigen labeled with a radioactive isotope, commonly squared to the power of 5 or cube H. Antibody, specific to the antigen, allowing selective binding. Separation system, separates bound antigen-antibody complexes from free antigen precipitation or adsorption methods. Radiation detector, measures radioactivity using a gamma counter for squared to the power of 5i or a scintillation counter for cube H. 2. Procedure of RIA preparation of radioactive antigen. The target antigen is labeled with a radioactive isotope, e.g., iodine-125 for protein hormones. Incubation with antibody. A fixed amount of antibody is mixed with a radioactive antigen tracer to form antigen-antibody complexes. Addition of an labeled sample antigen. A sample containing an unknown concentration of the antigen is added. The unlabeled antigen competes with the labeled antigen for binding to the antibody. Separation of bound and free antigen. Precipitation method. Addition of a second antibody or polyethylene glycol to precipitate bound complexes. Adsorption method. Activated charcoal or solid phase separation is used. Measurement of radioactivity. The radioactivity in the bound fraction is measured using a gamma counter for squared to the power of 5i or a beta counter for cube H. The amount of radioactivity is inversely proportional to the concentration of the sample antigen. Standard curve and quantification. A standard curve is generated using known antigen concentrations. The sample concentration is determined by comparing its radioactivity to the standard curve. 3. Types of RIA. Competitive RIA. Labeled and unlabeled antigens compete for antibody binding. Higher sample antigen right pointing arrow less binding with the radioactive antigen right pointing arrow lower measured radioactivity. Non-competitive RIA. Immunometric RIA. Uses two antibodies sandwich method to directly measure the antigen. Used for large molecules like proteins and hormones. IV. Applications of RIA medical diagnostics. Hormone assays, insulin, thyroid hormones, cortisol, growth hormone. Infectious disease testing, hepatitis, HIV. Tumor markers, alpha-fetoprotein, carcinoembryonic antigen. Pharmacology and drug monitoring. Measures drug levels in blood, e.g., therapeutic monitoring of antibiotics, narcotics, and steroids. Used in toxicology for drug screening. Biomedical research. Studies antigen-antibody interactions. Quantifies biomolecules in cell culture and experimental studies. Food and environmental analysis. Detection of contaminants, toxins, and pesticides in food and water. The advantages and limitations of RIA. Advantages, limitations. Extremely high sensitivity detects picogram levels. Use of radioactive materials requires strict regulations. High specificity due to antigen-antibody interactions. Short shelf life of radioactive tracers. Quantitative and reproducible results require specialized equipment. Gamma counter can measure molecules in complex biological samples. You said. No tables, just paragraph. ChatGPT said. Radioimmunoassay, RIA. Radioimmunoassay, RIA, is a highly sensitive technique used for the quantitative measurement of biomolecules such as hormones, proteins, and drugs. It is based on the principle of antigen-antibody interactions and utilizes a radioactive isotope labeled antigen to compete with an unlabeled antigen for antibody binding. This competitive binding mechanism allows the precise detection of analytes in biological samples. Developed by Rosalind Yalow and Solomon Burson in the 1950s, RIA revolutionized biomedical research and clinical diagnostics by enabling the detection of minute concentrations of substances in blood, serum, and other fluids. The fundamental principle of RIA involves competition between a known quantity of radioactively labeled antigen and an unknown amount of unlabeled antigen present in the sample. Both forms of the antigen bind to a fixed amount of specific antibody, forming antigen-antibody complexes. As the concentration of the unlabeled antigen increases, it displaces the labeled antigen from binding to the antibody. After the reaction reaches equilibrium, the antigen-antibody complexes are separated from the unbound components using precipitation or adsorption techniques. The radioactivity of the bound fraction is then measured using a gamma counter for isotopes such as iodine-125 or a beta counter for isotopes like tritium. The amount of radioactivity detected is inversely proportional to the concentration of the unlabeled antigen in the sample, allowing for precise quantification using a standard curve. The RIA procedure involves several key steps. First, a radioactive antigen is prepared by labeling the target molecule with a suitable isotope. This labeled antigen is then mixed with a specific antibody, allowing the formation of antigen-antibody complexes. Next, the sample containing the unknown concentration of the unlabeled antigen is introduced, initiating the competitive binding process. After incubation, a method such as precipitation or adsorption is used to separate the bound antigen-antibody complexes from the free antigen. Finally, the radioactivity of the bound fraction is measured, and the concentration of the antigen in the sample is determined by comparing it to a standard calibration curve. There are different types of RIA, with the most common being competitive RIA, in which labeled and unlabeled antigens compete for a limited number of antibody binding sites. In this method, higher concentrations of a sample antigen result in less binding of the labeled antigen, leading to a decrease in measured radioactivity. Another variant is the immunometric or non-competitive RIA, where two antibodies are used in a sandwich assay format to directly capture and measure the antigen. This type is particularly useful for detecting larger biomolecules such as hormones and proteins. RIA has extensive applications in medical diagnostics, pharmacology, and research. It is widely used to measure hormone levels, including insulin, thyroid hormones, cortisol, and growth hormone, providing critical information for diagnosing endocrine disorders. It is also employed in detecting tumor markers such as alpha-fetoprotein and carcinoembryonic antigen, aiding in cancer diagnosis and monitoring. In pharmacology, RIA is used to measure drug levels in blood, ensuring proper therapeutic dosages and monitoring drug metabolism. It is also valuable in toxicology for detecting narcotics and steroids in biological samples. Additionally, RIA is used in food safety and environmental testing to detect contaminants, toxins, and pesticides. Despite its high sensitivity and specificity, RIA has limitations. The use of radioactive isotopes poses safety hazards and requires strict regulatory compliance. The short half-life of certain isotopes limits the shelf life of reagents, necessitating careful handling and storage. Specialized equipment such as gamma counters is required for radioactivity measurement, increasing operational costs. Moreover, the technique involves multiple incubation and separation steps, making it labor-intensive compared to newer immunoassay techniques such as enzyme-linked immunosorbent assay ELISA. In conclusion, radioimmunoassay remains one of the most precise and sensitive methods for detecting and quantifying biomolecules. Although its use has declined with the development of non-radioactive immunoassays, it continues to be a valuable tool in specialized research and clinical applications where ultra-sensitive detection is required. You said, Western blot. ChatGPT said, Western blot. Western blot, also known as immunoblotting, is a widely used laboratory technique for detecting and quantifying specific proteins in a complex mixture. It is based on the principle of protein separation by gel electrophoresis, followed by transfer to a membrane and detection using specific antibodies. This method provides high specificity and sensitivity, making it a valuable tool in molecular biology, biochemistry, and medical diagnostics. The procedure of Western blot involves several key steps. First, proteins are extracted from a biological sample such as tissue or cultured cells and are denatured using detergents like sodium dodecyl sulfate (SDS). The denatured proteins are then separated based on their molecular weight using SDS polyacrylamide gel electrophoresis (SDS page. The negatively charged proteins migrate through the gel under an electric field, with smaller proteins moving faster than larger ones, leading to effective size-based separation. Once separated, the proteins are transferred from the gel onto a solid membrane, typically made of nitrocellulose or polyvinylidene difluoride (PVDF). This transfer is achieved through electroblotting, where an electric current drives the proteins from the gel onto the membrane. The membrane acts as a stable platform for protein detection. To prevent non-specific binding of antibodies, the membrane is then blocked using a protein-rich solution such as bovine serum albumin (BSA) or non-fat milk. Protein detection in Western blot relies on the use of specific antibodies. A primary antibody, which recognizes the target protein, is first incubated with the membrane. After washing off unbound antibodies, a secondary antibody conjugated to detection enzymes such as horseradish peroxidase (HRP) or alkaline phosphatase (AP) is applied. This secondary antibody binds to the primary antibody and allows visualization of the target protein. Detection is typically performed using chemiluminescence, fluorescence, or colorimetric methods, depending on the labeling of the secondary antibody. In chemiluminescent detection, enzyme-substrate reactions produce light, which is captured using an imaging system. Fluorescent detection uses labeled antibodies that emit light upon excitation, while colorimetric detection generates a visible pigment for manual observation. Western blot has numerous applications in research and diagnostics. It is commonly used for detecting protein expression in cells and tissues, verifying the presence of specific proteins, and studying post-translational modifications such as phosphorylation. In medical diagnostics, Western blot plays a crucial role in confirming infections such as HIV and Lyme disease by detecting pathogen-specific antibodies in patient samples. It is also used in cancer research to analyze oncogenic protein expression and signaling pathways. Despite its advantages, Western blot has some limitations. It is a time-consuming and labor-intensive process that requires multiple steps, including careful optimization of antibody concentrations and washing conditions to reduce background noise. The technique also requires expertise to interpret results accurately, as protein bands may sometimes be faint or show non-specific binding. Additionally, the method is semi-quantitative, meaning that while it provides relative protein abundance, it is not as precise as mass spectrometry-based proteomic techniques. The technique also requires expertise to interpret results accurately, as protein bands may sometimes be faint or show non-specific binding. Additionally, the method is semi-quantitative, meaning that while it provides relative protein abundance, it is not as precise as mass spectrometry-based proteomic techniques. In conclusion, Western blot remains a powerful and widely used technique for protein analysis. Its high specificity and ability to detect proteins in complex mixtures make it indispensable in molecular biology, biomedical research, and clinical diagnostics. While newer technologies such as multiplex assays and mass spectrometry are emerging, Western blot continues to be a reliable method for protein detection and characterization. You said. Immunoprecipitation. ChatGPT said. Immunoprecipitation (IP). Immunoprecipitation (IP) is a widely used biochemical technique that isolates a specific protein from a complex mixture using an antibody that specifically binds to that protein. This technique is essential for studying protein interactions, post-translational modifications, and protein expression levels in various biological samples. By using antibodies to selectively capture a target protein, IP enables researchers to analyze protein functions in cellular and molecular processes. The immunoprecipitation process begins with the preparation of a protein extract from a biological sample, such as cultured cells or tissue lysates. The proteins are solubilized using detergents or buffers to ensure efficient antibody binding. A specific antibody against the target protein is then added to the lysate, allowing antigen-antibody complexes to form. To facilitate the separation of these complexes, protein binding beads, such as protein A, protein G, or protein L-coated agros or magnetic beads, are introduced. These beads have a strong affinity for the Fc region of the antibody, enabling the capture of antibody-protein complexes. After incubation, the antibody-bound proteins are separated from the rest of the sample using centrifugation for agros beads or magnetic separation for magnetic beads. The beads are washed multiple times to remove non-specifically bound proteins. The target protein can then be eluted from the beads using a denaturing buffer, typically containing SDS and reducing agents, making it suitable for further analysis by Western blot, mass spectrometry, or enzymatic assays. Several variations of immunoprecipitation exist, each designed for specific experimental objectives. Classic or standard IP is used to isolate and study a single protein from a complex lysate. Co-immunoprecipitation company IP allows the identification of protein-protein interactions by isolating a protein along with its binding partners. Chromatin immunoprecipitation chip is used to study DNA-protein interactions, such as transcription factor binding to specific DNA regions. Ribonucleoprotein immunoprecipitation (RIP) is similar to chip but focuses on RNA-protein interactions. Tag protein immunoprecipitation involves the use of epitope tagged recombinant proteins, e.g., FLAG, HA, MYC, GFP, for easy detection and purification. Immunoprecipitation is extensively used in research to study protein interactions, identify binding partners, and examine post-translational modifications such as phosphorylation, ubiquitination, or glycosylation. It is also employed in biomedical research and diagnostics, such as detecting viral proteins, analyzing immune complexes in autoimmune diseases, and studying cell signaling pathways. Despite its utility, immunoprecipitation has some limitations. The success of the technique relies heavily on the specificity and affinity of the antibody used, as non-specific binding can lead to false positives. Additionally, harsh elution conditions can sometimes disrupt protein complexes, making it challenging to study weak or transient interactions. Ensuring proper controls, such as using an irrelevant antibody isotype control or performing an experiment without the primary antibody, no antibody control, is essential to validate results. In conclusion, immunoprecipitation is a powerful method for protein isolation and interaction studies. Its versatility allows researchers to investigate complex cellular processes, making it a fundamental technique in molecular biology, proteomics, and biomedical research. With advancements in antibody technology and improved detection methods, IP continues to be an essential tool for understanding protein function and cellular regulation. You said. Flow cytometry. ChatGPT said. Flow cytometry. Flow cytometry is a powerful technique used to analyze and sort cells or particles based on their physical and biochemical properties as they pass through a laser beam in a fluid stream. This method enables rapid, high throughput, and quantitative analysis of cell populations, making it widely used in immunology, microbiology, cancer research, and clinical diagnostics. The fundamental principle of flow cytometry involves suspending cells or particles in a liquid medium and passing them through a narrow nozzle, where they move in a single file stream. A laser beam illuminates each particle, and detectors measure the scattered light and emitted fluorescent signals. Forward scatter FSC measures cell size, while side scatter SSC provides information about internal complexity or granularity. Additionally, fluorescent signals from labeled antibodies or dyes provide information on specific cellular markers. One of the key advantages of flow cytometry is its ability to detect multiple parameters simultaneously. Fluorescently labeled antibodies targeting specific surface or intracellular proteins can be used to distinguish different cell types within a heterogeneous population. Fluorophores such as fluorescein isothiocyanate, FITC, phycoerythrin PD, and L-phycocyanin APC emit distinct fluorescent signals, which are captured by multiple detectors, allowing multicolor analysis. The process of flow cytometry typically includes several steps. First, cells are isolated and suspended in a suitable buffer to maintain their viability. The sample is then stained with fluorophore conjugated antibodies or dyes specific to the markers of interest. After staining, the cells are washed to remove excess reagents and then analyzed in a flow cytometer. As each cell passes through the laser, light scatter and fluorescent signals are detected and converted into electronic signals. These data are processed by specialized software to generate histograms, dot plots, or contour plots for analysis. One of the most advanced applications of flow cytometry is fluorescence-activated cell sorting (FACS), which allows the physical separation of cells based on their fluorescence characteristics. In FACS, charged droplets containing individual cells are deflected by an electric field into different collection tubes, enabling the isolation of specific cell subpopulations for further experiments. Flow cytometry has a wide range of applications in research and medicine. It is extensively used in immunophenotyping to characterize immune cell populations, such as distinguishing T cells, B cells, and natural killer cells based on their surface markers. In cancer diagnostics, it helps in detecting abnormal cell populations, such as leukemia and lymphoma cells. It is also applied in apoptosis assays to study programmed cell death, cell cycle analysis to determine DNA content and proliferation rates, and microbial detection for identifying bacteria and fungi in clinical and environmental samples. Additionally, it is used in vaccine development, stem cell research, and quality control in biopharmaceutical production. Despite its numerous advantages, flow cytometry has some limitations. The technique requires expensive instrumentation and specialized expertise for data analysis and interpretation. Additionally, sample preparation must be carefully optimized to avoid cell clumping, autofluorescence, and non-specific binding of antibodies. The high sensitivity of flow cytometry can also lead to background noise, requiring appropriate controls, such as unstained samples, single stain controls, and fluorescence -1 FMO controls to ensure accurate results. In conclusion, flow cytometry is an indispensable tool for cellular and molecular analysis. Its ability to provide rapid, multi-parameter analysis at a single cell level makes it a critical technique in both basic research and clinical diagnostics. With ongoing advancements in spectral flow cytometry and machine learning-based data analysis, flow cytometry continues to expand its applications in precision medicine, immunotherapy, and biotechnology. You said. Immunofluorescence microscopy, ChatGPT said. Immunofluorescence microscopy. Immunofluorescence microscopy is a powerful technique used to visualize specific proteins or cellular structures in biological samples using fluorescently labeled antibodies. It is widely employed in cell biology, immunology, and pathology to study protein localization, cell signaling pathways, and disease-related biomarkers. This method provides high specificity and sensitivity, enabling the detailed visualization of cellular components at the subcellular level. The principle of immunofluorescence microscopy relies on the ability of antibodies to bind specifically to their target antigens. These antibodies are conjugated to fluorescent dyes, known as fluorophores, which emit light at specific wavelengths when excited by light source, such as mercury or LED lamp in a fluorescence microscope. The emitted fluorescence is captured using specialized filters, allowing researchers to observe the distribution of target proteins in cells or tissues. There are two main types of immunofluorescence: direct immunofluorescence (DIF) and indirect immunofluorescence (IIF). In direct immunofluorescence, a fluorophore is directly conjugated to the primary antibody, which binds to the target antigen. This method is simple and produces minimal background noise but has lower signal amplification. Indirect immunofluorescence, on the other hand, involves using an unlabeled primary antibody to bind to the antigen, followed by a fluorophore conjugated secondary antibody that recognizes the primary antibody. This approach enhances signal intensity due to multiple secondary antibodies binding to a single primary antibody, increasing detection sensitivity. The immunofluorescence protocol involves several key steps. First, the cells or tissue sections are fixed using formaldehyde or methanol to preserve cellular structures. The sample is then permeabilized using detergents like Triton X100 to allow antibodies to access intracellular antigens. After blocking non-specific binding sites with a protein-rich solution, such as bovine serum albumin or normal serum, the sample is incubated with the primary antibody. Following thorough washing, the appropriate fluorophore conjugated secondary antibody is applied. Finally, the sample is mounted on a glass slide with an anti-fade reagent to prevent photobleaching and is examined under a fluorescence microscope. Immunofluorescence microscopy has numerous applications in biological and medical research. It is widely used in cellular localization studies to determine the subcellular distribution of proteins, such as identifying nuclear, cytoplasmic, or membrane-associated proteins. It plays a crucial role in disease diagnostics, particularly in detecting autoimmune diseases, viral infections, and cancer biomarkers. For example, in dermatopathology, direct immunofluorescence is used to diagnose autoimmune blistering disorders by detecting immune complex deposition in skin biopsies. In neuroscience, immunofluorescence helps in mapping neural circuits and identifying neurotransmitter-specific neurons. It is also applied in virology and microbiology to detect pathogens in infected tissues. Despite its advantages, immunofluorescence microscopy has some limitations. Fluorophores are susceptible to photobleaching, which can reduce signal intensity over time. Additionally, non-specific antibody binding can produce background fluorescence, requiring careful optimization of antibody dilutions and blocking conditions. Another limitation is the resolution constraint imposed by conventional light microscopy, which prevents visualization of structures smaller than the diffraction limit, approximately 200 nanometers. However, advances in super-resolution microscopy techniques, such as stimulated emission depletion (STED) microscopy, structured illumination microscopy (SIM), and stochastic optical reconstruction microscopy (STORM) have overcome this limitation, allowing for nanometer scale imaging of cellular structures. In conclusion, immunofluorescence microscopy remains a fundamental tool in biomedical research for studying protein expression, cellular architecture, and disease mechanisms. Its ability to provide high-resolution, specific, and multicolor imaging makes it invaluable for understanding complex biological processes. With continuous advancements in fluorophore technology and imaging systems, immunofluorescence microscopy continues to evolve, offering deeper insights into cellular and molecular biology. You said. Detection of molecules in living cells CSIR lecture from now on focus on previously questioned cane areas and points and add 2 or 3 previous year's questions and answers year in bracket. ChatGPT said. Memory updated. Detection of molecules in living cells. The detection of molecules within living cells is essential for understanding cellular processes, molecular interactions, and signaling pathways. Various techniques allow researchers to track biomolecules in real time, providing insights into cellular function, dynamics, and responses to environmental stimuli. These techniques are widely used in molecular and cell biology, drug discovery, and disease diagnostics. Key techniques for molecule detection in living cells: fluorescence microscopy and fluorescent probes. Fluorescent dyes e.g., DAPI, FITC, rhodamine, and genetically encoded fluorophores e.g., GFP, RFP, McCarry are used to visualize molecules within live cells. Fluorescence recovery after photobleaching (FRAP) and fluorescence loss in photobleaching (FLIP) help study molecular dynamics and diffusion within cells. Fluorescence resonance energy transfer (FRET) detects molecular interactions by measuring energy transfer between two fluorophores in close proximity. Live cell imaging with confocal and super resolution microscopy. Confocal microscopy provides high resolution, real-time imaging of fluorescently labeled molecules in living cells. Super resolution techniques like STED, POM, and STORM overcome diffraction limits, allowing nanoscale visualization of molecules. Biosensors and reporter systems. Genetically encoded biosensors e.g., calcium-sensitive GCAMP, ATP sensors help monitor intracellular metabolites and signaling molecules. Luciferase-based reporter assays track gene expression and protein activity in living cells. Single molecule imaging techniques. Total internal reflection fluorescence (TIRF) microscopy enables visualization of single molecules near the cell membrane. Single molecule tracking (SMT) techniques analyze molecular movement and interactions in real time. Flow cytometry and fluorescence activated cell sorting (FACS). Flow cytometry detects and quantifies labeled molecules within cells using fluorescent markers. FACS isolates specific cell populations based on fluorescence signals for further analysis. Raman spectroscopy and infrared IR microscopy. Raman and IR spectroscopy detect molecular vibrations, allowing label-free analysis of biomolecules in live cells. Surface-enhanced Raman scattering (SERS) enhances detection sensitivity for trace biomolecules. Genetically encoded tags for protein tracking. Fusion proteins with GFP or other fluorescent tags allow tracking of specific proteins in living cells. Halotag and SnapTag systems provide alternative labeling strategies for dynamic imaging. Previously asked CSIRNet question CSIRNET 2021. Which technique is commonly used to study protein-protein interactions in living cells using fluorescence energy transfer? A. Pret. B. Prep. C. Western blot. D. ELISA. Answer. A. Pret. CSIRNET 2019. Which of the following methods allows real-time tracking of a single molecule inside a living cell? A. Turf microscopy. B. Electron microscopy. C. ELISA. D. Mass spectrometry. Answer. A. Turf microscopy. CSIRNET 2017. Which of the following is a genetically encoded biosensor used for detecting calcium levels in live cells? A. GCamp. B. GFP. C. Rhodamine. B. T. Luciferase. Answer. A. GCamp. By integrating advanced imaging, biosensors, and spectroscopy-based techniques, researchers can obtain real-time insights into molecular functions in living cells, making these methods invaluable in modern biological research. You said. In situ localization by techniques. ChatGPT said. In situ localization by techniques. In situ localization refers to the detection and visualization of specific molecules, such as nucleic acids or proteins, within their native cellular or tissue environment. This approach is crucial for understanding spatial gene expression, protein distribution, and molecular interactions in cells and tissues. Various techniques have been developed to study in situ localization with high specificity and resolution, aiding research in developmental biology, pathology, and molecular diagnostics. Key techniques for in situ localization: in situ hybridization (ISH). ISH is used for detecting specific DNA or RNA sequences within cells or tissue sections. A label complementary nucleic acid probe hybridizes to the target sequence, allowing localization of specific genes or transcripts. Types of ISH. Fluorescence in situ hybridization. FISH uses fluorescent probes to detect nucleic acids, commonly applied in chromosome mapping and detecting genetic abnormalities. Chromogenic in situ hybridization. CISH uses enzyme-linked probes that produce a color reaction visible under a light microscope. Immunohistochemistry (IHC) and immunocytochemistry (ICC). IHC is used to detect proteins in tissue sections, while ICC is used for cultured cells. Antibodies specific to the target protein are used, followed by detection using enzymatic, e.g., HRP-DAB or fluorescence-based methods. IHC is widely used in cancer diagnostics and histopathology to study protein expression patterns. In situ PCR involves amplification of specific DNA or RNA sequences directly within fixed cells or tissues. Enhances sensitivity for detecting low-abundance nucleic acids while maintaining spatial resolution. Used in detecting viral infections, oncogenes, and rare mutations in tissues. RNA-scope technology. A highly sensitive variation of ISH used for detecting single RNA molecules in tissues. Provides better resolution than conventional ISH and enables multiplex detection of multiple transcripts. Commonly used in cancer research and neuroscience to study gene expression heterogeneity. Autoradiography. Uses radiolabel probes to detect DNA, RNA, or proteins in cells or tissues. After hybridization or binding, the sample is exposed to an X-ray film to visualize signal localization. Previously used in molecular biology research but has been largely replaced by fluorescent and chromogenic methods. Mass spectrometry imaging (MSI) allows spatial mapping of biomolecules such as proteins, lipids, and metabolites within tissue sections. Provides high-resolution molecular analysis without requiring antibodies or probes. Applied in cancer biomarker discovery and pharmacokinetic studies. Confocal and super-resolution microscopy for localization. Confocal laser scanning microscopy enhances in situ localization by providing high-resolution optical sectioning of samples. Super-resolution techniques like STED and POM enable nanoscale imaging of molecules in their cellular context. Previously asked CSIRNet question CSIRNET 2022. Which of the following techniques is commonly used to detect mRNA expression in tissue sections? Immunohistochemistry. B. Fluorescence in situ hybridization. FISH. C. Western blot. D. SDS page. Answer. B. Fluorescence in situ hybridization. FISH. CSIRNET 2020. What is the primary advantage of in situ PCR compared to conventional PCR? A. Higher specificity. B. Spatial localization of amplified sequences. C. Faster amplification process. D. Does not require primers. Answer. B. Spatial localization of amplified sequences. CSIRNET 2018. Which technique allows direct visualization of protein localization in intact tissue sections? A. SDS page. B. Immunohistochemistry. IHC. C. ELISA. D. Southern blot. Answer. B. Immunohistochemistry. IHC. In situ localization techniques provide essential insights into molecular organization and function in biological systems. These methods continue to evolve, offering improved sensitivity and specificity for studying gene and protein expression in their native environments. You said. FISH. ChatGPT said. Fluorescence in situ hybridization. FISH. Fluorescence in situ hybridization. FISH is a powerful molecular cytogenetic technique used to detect and localize specific nucleic acid sequences within cells and tissue samples. It utilizes fluorescently labeled probes that bind to complementary DNA or RNA sequences, allowing visualization under a fluorescence microscope. FISH is widely used in genetics, cancer diagnostics, microbial identification, and gene mapping. Principle of FISH. FISH involves the hybridization of a fluorescently labeled DNA or RNA probe to a specific target sequence in fixed cells or tissues. The unbound probe is washed away, and the hybridized signal is detected using fluorescence microscopy. The technique enables the detection of gene amplifications, deletions, chromosomal translocations, and aneuploidy. Steps in FISH procedure: sample preparation. Cells or tissue sections are fixed to preserve morphology and nucleic acid integrity. Chromosomes are denatured to make the DNA single-stranded for probe binding. Probe labeling. Probes are synthesized to be complementary to the target sequence. Labeled with fluorophores such as FITC, Texas Red, or Cy3. Hybridization. The labeled probe is applied to the sample and incubated under controlled conditions to allow binding to the target sequence. Washing and counterstaining. Unbound probes are washed off to reduce background fluorescence. DAPI, 4-pete, 6-diamidino-2 phenylengyl, or other counterstains are used to visualize cell nuclei. Fluorescence microscopy. The sample is observed under a fluorescence microscope, where different fluorophores emit distinct signals, enabling detection of specific sequences. Types of FISH chromosome painting. Entire chromosomes or chromosomal regions are labeled with probes to study structural abnormalities. Centromeric FISH, CEN-FISH. Detects centromere regions to identify aneuploidy or chromosomal instability. Telomere FISH. Analyzes telomere length and organization, useful in aging and cancer studies. RNA FISH. Detects specific RNA molecules within cells to study gene expression patterns. Multiplex FISH, M-FISH. Uses multiple fluorophores to simultaneously detect multiple chromosomal regions, allowing comprehensive genome analysis. Applications of FISH. Genetic disorders. Detects chromosomal abnormalities such as Down syndrome, trisomy 21, Turner syndrome, and deletions associated with genetic diseases. Cancer diagnosis. Identifies gene amplifications, e.g., HER2 in breast cancer translocations, e.g., BCR-ABL in chronic myeloid leukemia, and other mutations. Microbial identification. Distinguishes bacterial and viral infections by targeting specific rRNA sequences. Evolutionary and comparative genomics. Helps in chromosome mapping and studying species relationships. Previously asked CSIR net question CSIR-NET 2021. Which of the following techniques is used to detect chromosomal translocations in cancer cells? A. ELISA. B. SDS page. C. Fluorescence in situ hybridization FISH D. Western blot. Answer. C. Fluorescence in situ hybridization FISH. CSIR-NET 2019. Which of the following is a major advantage of FISH over conventional karyotyping? A. Requires metaphase chromosomes for analysis. B. Can detect small genetic changes undetectable by karyotyping. C. Does not require fluorescent probes. D. Cannot be used for RNA detection. Answer. B. Can detect small genetic changes undetectable by karyotyping. CSIR-NET 2017. Which fluorescent dye is commonly used to counterstain nuclei in FISH experiments? A. Kumasi blue. B. DAPI. C. Ethylene bromide. D. Bromophenol blue. Answer. B. DAPI. FISH remains a crucial technique in molecular biology, genetics, and medical diagnostics, enabling precise and rapid detection of chromosomal and genetic abnormalities. You said, GISH. ChatGPT said. Genomic in situ hybridization, GISH. Genomic in situ hybridization, GISH, is a molecular cytogenetic technique used to distinguish and visualize entire genomes within hybrid organisms, polyploids, or interspecific hybrids. It employs whole genomic DNA from one species as a probe to hybridize with chromosomes of another species, allowing the identification of parental genome contributions in hybrids. GISH is widely applied in plant and animal genetics, evolutionary biology, and breeding programs. Principle of GISH. GISH is based on the hybridization of total genomic DNA from one species used as a probe onto the chromosomes of a hybrid or polyploid species. The probe is labeled with fluorophore, enabling visualization under a fluorescence microscope. By comparing the hybridization patterns, researchers can differentiate between the parental genomes and study chromosome behavior in hybrids. Steps in GISH procedure: chromosome preparation. Root tips, meristematic tissues, or other dividing cells are fixed and prepared for chromosome spreading on slides. Chromosomes are denatured to make DNA single-stranded for probe binding. Probe preparation. Genomic DNA is isolated from one of the parent species and fragmented. Labeled with fluorophores, e.g., Cy3, FITC, or biotin HRP for visualization. In some cases, blocking DNA from another parental genome is added to reduce non-specific binding. Hybridization. The label probe is applied to the chromosome spread and incubated under controlled conditions. Hybridization occurs between complementary DNA sequences. Post-hybridization washing. Unbound probes are removed by washing with buffers to enhance specificity. Detection and visualization. Fluorescence microscopy is used to detect hybridized signals. Different parental genomes appear in distinct colors, allowing identification of genomic origin. Applications of GISH. Hybrid identification and parental genome differentiation. Used to confirm hybridization events in plants and animals. Differentiates chromosomes of different species within hybrid organisms. Polyploidy studies. Helps in understanding genome composition in allopolyploids, organisms with genomes from different species. Used in crops like wheat, cotton, and brassica to analyze hybrid origins. Chromosome evolution and integration analysis. Detects chromosome exchanges and interactions between species. Provides insights into chromosomal evolution in plant and animal lineages. Genome organization and speciation studies. Reveals chromosomal rearrangements and genome stability in hybrid species. Helps study evolutionary relationships between closely related species. Comparison: GISH versus FISH. FISH uses specific DNA probes to detect particular genes or chromosomal regions, whereas GISH uses total genomic DNA to distinguish entire genomes. FISH is commonly used for gene mapping, chromosomal abnormalities, and diagnostics, while GISH is primarily used for genome differentiation in hybrids and polyploids. Previously asked CSIR net question CSIR-NET 2021. Which cytogenetic technique is best suited to distinguish parental genomes in an interspecific hybrid? A. Western blot. B. Genomic in situ hybridization GISH. C. ELISA. D. Southern blot. Answer. B. Genomic in situ hybridization GISH. CSIR-NET 2018. What is the main difference between FISH and GISH? A. FISH detects single genes, whereas GISH differentiates entire genomes. B. FISH is only used in plants, while GISH is used in animals. C. GISH does not require label probes. D. FISH is used for RNA detection, whereas GISH is used for proteins. Answer. A. FISH detects single genes, whereas GISH differentiates entire genomes. CSIR-NET 2016. Which of the following techniques would be useful for detecting genome composition in an allopolyploid species? A. GISH. B. Northern blot. C. Western blot. D. PCR. Answer. A. GISH. Genomic in situ hybridization GISH remains an essential tool in plant breeding, hybrid identification, and evolutionary biology, offering precise genome differentiation in polyploid and hybrid species. You said. Biophysical method. ChatGPT said. Biophysical methods in molecular biology. Biophysical methods are instrumental in studying the structural and functional properties of biomolecules, including proteins, nucleic acids, and macromolecular complexes. These techniques help elucidate molecular interactions, conformational changes, and dynamics, which are essential for understanding biological processes at a mechanistic level. Common biophysical methods: 1. X-ray crystallography. Used to determine the atomic structure of proteins, DNA, and RNA. Crystals of biomolecules are exposed to X-rays, and the diffraction pattern is analyzed to construct a 3D structure. Essential for drug discovery and understanding enzyme mechanisms. Requires high purity crystals, which can be a limitation. 2. Nuclear magnetic resonance (NMR) spectroscopy studies the structure and dynamics of biomolecules in solution. Relies on the magnetic properties of atomic nuclei, such as hydrogen and carbon. Useful for studying protein-ligand interactions, protein folding, and conformational changes. Limited to small and medium-sized proteins, usually below 50 kDa. 3. Circular dichroism (CD) spectroscopy. Measures the secondary structure of proteins and nucleic acids based on their interaction with circularly polarized light. Differentiates between alpha helices, beta sheets, and random coils. Useful for monitoring protein folding, stability, and conformational changes. 4. Surface plasmon resonance (SPR) used to study real-time biomolecular interactions without labeling. Measures changes in refractive index when molecules bind to a sensor chip. Quantifies binding kinetics, affinity, and specificity of interactions. Widely used in drug discovery for evaluating protein-ligand interactions. 5. Isothermal titration calorimetry (ITC). Measures heat changes during molecular interactions, providing insights into binding affinity, enthalpy, and entropy. Useful for studying enzyme kinetics, protein-ligand interactions, and thermodynamic properties. Requires high concentrations of pure biomolecules. 6. Differential scanning calorimetry (DSC). Measures thermal stability and conformational changes in biomolecules by detecting heat capacity changes. Used for protein folding studies, stability testing, and drug formulation. 7. Single molecule fluorescence spectroscopy examines the behavior of individual molecules rather than ensemble averages. Techniques include fluorescence resonance energy transfer (FRET) and fluorescence correlation spectroscopy (FCS). Used for studying molecular dynamics, conformational changes, and biomolecular interactions. 8. Mass spectrometry (MS). Analyzes molecular mass and composition of biomolecules. Used in proteomics for protein identification and post-translational modifications. Includes techniques like MALDI-TOF and SCMS for high-resolution analysis. 9. Atomic force microscopy (AFM). Provides high-resolution imaging of biomolecules at the nanoscale. Measures molecular interactions and mechanical properties of biomolecules. 10. Cryo-electron microscopy (cryo-EM). Uses electron beams to determine the structure of large biomolecular complexes without requiring crystallization. Ideal for studying membrane proteins and dynamic molecular assemblies. Revolutionized structural biology, especially for large macromolecules like ribosomes and viruses. Applications of biophysical methods. Structural biology, understanding the 3D structure of proteins, DNA, and RNA. Drug discovery, identifying molecular targets and studying drug receptor interactions. Enzyme kinetics, investigating catalytic mechanisms and reaction rates. Protein-protein interactions, exploring molecular recognition and signaling pathways. Molecular dynamics, studying conformational changes and folding mechanisms. Previously asked CSIR net question CSIR-NET 2020. Which biophysical technique is used to determine the atomic structure of proteins by analyzing X-ray diffraction patterns? A. NMR spectroscopy. B. X-ray crystallography. C. Surface plasmon resonance. SPRT. Atomic force microscopy. AFM. Answer. B. X-ray crystallography. CSIR-NET 2018. Which of the following techniques is used to measure the thermal stability of proteins? A. Circular dichroism. C. Deep spectroscopy. B. Differential scanning calorimetry. DSC. C. Surface plasmon resonance. SPRT. Mass spectrometry. MS. Answer. B. Differential scanning calorimetry. DSC. CSIR-NET 2016. Which technique can be used to study real-time biomolecular interactions without requiring labels? A. Isothermal titration calorimetry. ITC. B. Surface plasmon resonance. SPR. C. Fluorescence microscopy. D. Mass spectrometry. Answer. B. Surface plasmon resonance. SPR. Biophysical methods provide essential insights into biomolecular structures and interactions, making them indispensable tools in molecular biology, biochemistry, and biomedical research. You said. Molecular analysis using UV. ChatGPT said. Molecular analysis using UV spectroscopy. Ultraviolet UV spectroscopy is a fundamental technique in molecular biology used to analyze nucleic acids, proteins, and other biomolecules. It is based on the absorption of ultraviolet light by molecules containing conjugated double bonds or aromatic rings. The primary application of UV spectroscopy in molecular biology is the quantification and purity assessment of nucleic acids and proteins. Principle of UV spectroscopy. UV spectroscopy relies on the Beer-Lambert law, which states that absorbance is directly proportional to the concentration of the absorbing substance. Molecules absorb UV light at specific wavelengths, depending on their electronic structure. The two major biomolecules analyzed using UV spectroscopy are nucleic acids, DNA, RNA, and proteins. UV spectroscopy for nucleic acid analysis 1. Quantification of DNA and RNA. Nucleic acids absorb UV light due to the purine adenine, guanine, and pyrimidine, cytosine, thymine, uracil bases. The maximum absorbance lambda max for DNA and RNA is at 260 nanometers, A260. The concentration of nucleic acids is determined using the formula. DNA concentration microgram per milliliter equals A260 times 50. RNA concentration microgram per milliliter equals A260 times 40. 2. Purity assessment of DNA and RNA. The ratio of absorbance at 260 nanometers to 280 nanometers, A260 is used to determine purity. A260 to 1.8 indicates pure DNA. A260 approximately 2.0 indicates pure RNA. A lower ratio suggests protein contamination due to protein absorbance at 280 nanometers. The A260 ratio is also checked for contaminants like phenol, carbohydrates, or salts. A ratio in GT 2.0 indicates high purity. UV spectroscopy for protein analysis 1. Protein quantification. Proteins absorb UV light due to the presence of aromatic amino acids tryptophan, tyrosine, and phenylalanine. The maximum absorbance for proteins occurs at 280 nanometers, A280. Protein concentration is calculated using A280 and an extinction coefficient specific to the protein. Alternatively, the Bradford, Lowry, and BCA assays use UV visible absorbance at different wavelengths for protein estimation. 2. Protein purity and contamination. The A260-A280 ratio helps identify nucleic acid contamination in protein samples. A260-A280 and GT 1.5 suggests nucleic acid contamination in a protein sample. Applications of UV spectroscopy in molecular biology. Nucleic acid quantification, used before PCR, sequencing, and cloning experiments. Protein quantification, essential for enzyme assays, electrophoresis, and Western blotting. Purity assessment, ensures high-quality DNA, RNA for molecular biology applications. Detection of structural changes, UV spectra can reveal protein denaturation or DNA-protein interactions. Previously asked CSIR net question CSIR-NET 2020. Which wavelength is used for nucleic acid quantification using UV spectroscopy? A. 280 nanometers. B. 230 nanometers. C. 260 nanometers. D. 320 nanometers. Answer. C. 260 nanometers. CSIR-NET 2018. If a DNA sample shows an A260-A280 ratio of 1.3, what does it indicate? A. Pure DNA sample. B. RNA contamination. C. Protein contamination. D. Salt contamination. Answer. C. Protein contamination. CSIR-NET 2016. Which aromatic amino acid contributes most to protein absorbance at 280 nanometers? A. Tyrosine. B. Tryptophan. C. Phenylalanine. D. Histidine. Answer. B. Tryptophan. UV spectroscopy remains a vital tool for molecular analysis, providing a rapid, non-destructive method for quantifying and assessing the purity of biomolecules. You said. Visible. ChatGPT said. Molecular analysis using visible spectroscopy. Visible spectroscopy, also known as colorimetric analysis, is widely used in molecular biology to quantify biomolecules, detect enzymatic reactions, and study biomolecular interactions. It is based on the absorption of visible light 400-700 nanometers by colored compounds or chromogenic reactions. Principle of visible spectroscopy. Like UV spectroscopy, visible spectroscopy follows the Beer-Lambert law, where absorbance is proportional to the concentration of the analyte. Some biomolecules naturally absorb visible light, while others require chromogenic or fluorogenic dyes for detection. Applications of visible spectroscopy in molecular biology 1. Protein quantification. Since proteins do not strongly absorb visible light, colorimetric assays are used. Bradford assay 595 nanometers uses Kumassi brilliant blue dye, which binds to proteins and shifts absorbance from 465 nanometers to 595 nanometers. Lowry assay 750 nanometers measures protein concentration based on copper ion reduction and a Folin-Choquette reagent reaction. Bisintronic acid BCA assay 562 nanometers uses bisintronic acid to detect copper ion reduction in proteins. 2. Nucleic acid quantification. DNA and RNA do not absorb visible light, but colorimetric dyes are used for detection. Diphenylamine assay detects DNA by forming a blue complex 595 nanometers. Orsinol assay detects RNA by forming a green complex 660 nanometers. 3. Enzyme activity assays. Enzymatic reactions producing colored products are monitored using visible spectroscopy. Alkaline phosphatase AP assay uses p-nitrophenyl phosphate PNPP, which turns yellow at 405 nanometers. Beta-galactosidase latch assay uses O-nitrophenyl beta-D-galactoguaranazide ONPG, forming a yellow product at 420 nanometers. Peroxidase-based assays use TMB3, 3-feet, 5-feet tetramethylbenzidine to generate a blue color at 450 nanometers. 4. Detection of metabolites. Many metabolic assays rely on chromogenic reactions for detection. Glucose estimation uses the glucose oxidase peroxidase God pod assay, forming a pink color at 540 nanometers. Lactate dehydrogenase LDH assay measures NADH reduction, monitored at 490 nanometers. 5. Colorimetric detection in ELISA. Enzyme-linked immunosorbent assay ELISA uses enzyme substrate reactions to produce colored products, typically measured at 450 nanometers TMB substrate or 405 nanometers PNPP substrate. Previously asked CSIR net question CSIR-NET 2021. Which protein estimation method uses Kumassi brilliant blue dye? A. Lowry assay. B. BCA assay. C. Bradford assay. D. ELISA. Answer: C. Bradford assay. CSIRNET 2019. Which enzyme is used in the colorimetric glucose estimation assay? A. Amylase. B. Glucose oxidase. C. Lactase. D. Peroxidase. Answer: B. Glucose oxidase. CSIRNET 2017. Which wavelength is commonly used to detect ELISA reactions using TMB substrate? A. 405 nanometers. B. 450 nanometers. C. 595 nanometers. D. 750 nanometers. Answer: B. 450 nanometers. Visible spectroscopy is a crucial tool in molecular biology for detecting and quantifying biomolecules, enzyme activities, and molecular interactions through colorimetric methods. You said. Fluorescence. ChatGPT said. Molecular analysis using fluorescence spectroscopy. Fluorescence spectroscopy is a powerful technique used in molecular biology to study biomolecules, detect specific proteins and nucleic acids, and analyze molecular interactions. It relies on the absorption of light at a specific wavelength and the subsequent emission of light at a longer wavelength. Principle of fluorescence spectroscopy. A fluorophore absorbs light at an excitation wavelength and emits light at a longer emission wavelength due to energy loss in the excited state. The difference between excitation and emission wavelengths is known as the Stokes shift. The intensity of fluorescence is proportional to the concentration of the fluorophore, allowing for sensitive detection. Applications of fluorescence spectroscopy in molecular biology. 1. Nucleic acid quantification. Fluorescent dyes enhance the sensitivity of DNA, RNA detection compared to UV spectroscopy. SYBR green and ethylene bromide (ETBR) interpolate between DNA bases and fluoresce upon UV excitation. Host and DAPI dyes bind to AT-rich regions of DNA and are used in cell imaging. 2. Protein quantification and analysis. Fluorescent protein labels, green fluorescent protein GFP, and variants like Macari and YFP allow real-time protein tracking. Fluorescent dyes, FITC, fluorescein, TRITC, rhodamine, and Alexa dyes conjugated to antibodies enable detection in Western blotting and immunofluorescence. Tryptophan fluorescence, intrinsic fluorescence of tryptophan residues in proteins can be used to study protein folding and conformational changes. 3. Fluorescence microscopy and imaging. Immunofluorescence microscopy uses fluorophore conjugated antibodies to visualize cellular components. Fluorescence in situ hybridization FISH detects specific DNA, RNA sequences in chromosomes using fluorescently labeled probes. Live cell imaging, fluorescent proteins and dyes enable real-time observation of cellular processes. 4. Flow cytometry. Cells labeled with fluorescent markers are analyzed based on light scattering and fluorescence intensity. Used for cell sorting, FACS, immune cell phenotyping, and apoptosis detection. 5. Fluorescence resonance energy transfer (FRET) measures interactions between biomolecules based on energy transfer between two fluorophores. Used in protein-protein interaction studies, biosensors, and molecular diagnostics. 6. Quantitative PCR, qPCR, and ELISA. Real-time PCR, qPCR uses SYBR green or Tachman probes to quantify nucleic acids. Fluorescence-based ELISA, more sensitive than colorimetric ELISA, detecting proteins with higher specificity. Previously asked CSIRNet question CSIRNET 2021. Which dye is commonly used for real-time PCR fluorescence detection? A. Ethidium bromide. B. SYBR green. C. DAPI. D. Rhodamine. Answer: B. SYBR green. CSIRNET 2019. Which fluorophore is used for live cell imaging? A. Hematoxylin. B. GFP. C. Crystal violet. D. Safranin. Answer: B. GFP. CSIRNET 2017. Which principle is used in FRET-based protein interaction studies? A. Energy transfer between two fluorophores. B. Light scattering by proteins. C. Absorbance at 280 nanometers. D. Nucleic acid hybridization. Answer: A. Energy transfer between two fluorophores. Fluorescence spectroscopy provides high sensitivity and specificity for molecular analysis, making it an essential tool in modern biological research. You said. Immunofluorescence microscopy. ChatGPT said. Immunofluorescence microscopy. Immunofluorescence microscopy is a powerful technique used to visualize specific proteins or cellular structures using fluorescently labeled antibodies. It is widely used in cell biology, pathology, and molecular diagnostics for detecting proteins in cells and tissues. Principle of immunofluorescence microscopy: a fluorophore conjugated antibody binds to a specific antigen in a sample. The sample is illuminated with light of a specific wavelength, exciting the fluorophore. The fluorophore emits light at a longer wavelength, which is detected under a fluorescence microscope. Different fluorophores FITC, TRITC, Alexa fluor, DAPI allow multicolor labeling of multiple targets in a single sample. Types of immunofluorescence microscopy. 1. Direct immunofluorescence DIF. A primary antibody is directly labeled with a fluorophore and binds to the target antigen. Advantages: faster, fewer steps, less background noise. Limitation: lower signal intensity due to fewer fluorophores per antigen. 2. Indirect immunofluorescence IIF. A primary antibody binds to the target antigen, and a secondary antibody conjugated to fluorophore binds to the primary antibody. Advantages: higher sensitivity due to multiple secondary antibodies binding to each primary antibody, amplifying the signal. Limitation: requires more steps, increasing the risk of background noise. Applications of immunofluorescence microscopy. 1. Cellular and subcellular protein localization detects specific proteins in cells or tissues, helping study cellular organization. Example: localization of tubulin, cytoskeleton, actin filaments, or nuclear proteins. 2. Disease diagnosis and pathology. Used in autoimmune disease detection, e.g., antinuclear antibodies, ANA, in lupus. Detection of viral infections like herpes simplex virus, HSV, in tissue samples. 3. Cancer research and biomarker detection identifies cancer-specific proteins in tissues, aiding in diagnosis and prognosis. Example: detection of HER2 in breast cancer or P53 in tumors. 4. Microbial and viral detection. Used to visualize bacterial, fungal, and viral antigens in clinical samples. Example: detection of mycobacterium tuberculosis or HIV proteins in infected cells. 5. Neuroscience research. Visualizes neurotransmitter receptors, synaptic proteins, and neuronal structures. Example: staining for GFAP, glial fibrillary acidic protein, to study astrocytes in the brain. Limitations of immunofluorescence microscopy: photobleaching. Fluorophores degrade over time with prolonged light exposure. Non-specific binding. Background fluorescence can interfere with interpretation. Requires specialized equipment. High-resolution fluorescence microscopes are expensive. Previously asked CSIRNet question CSIRNET 2021. Which of the following is a major advantage of indirect immunofluorescence over direct immunofluorescence? A. Reduced background fluorescence. B. Higher sensitivity due to signal amplification. C. Faster detection time. D. No need for secondary antibodies. Answer: B. Higher sensitivity due to signal amplification. CSIRNET 2019. Which fluorophore is commonly used for nuclear staining in immunofluorescence microscopy? A. FIT. B. Rhodamine. C. DAPI. D. Alexa fluor 488. Answer: C. DAPI. CSIRNET 2017. Immunofluorescence microscopy is commonly used to detect which of the following? A. DNA methylation patterns. B. Membrane potential of mitochondria. C. Localization of proteins within cells. D. RNA polymerase activity. Answer: C. Localization of proteins within cells. Immunofluorescence microscopy is a highly specific and sensitive method for visualizing molecular components in biological samples, making it invaluable for research and diagnostics. You said. Detection of molecules in living cells. ChatGPT said. Detection of molecules in living cells. The detection of biomolecules in living cells is crucial for understanding cellular functions, signaling pathways, and disease mechanisms. Various techniques allow real-time monitoring of proteins, nucleic acids, metabolites, and ions within living cells without disrupting their physiological state. Key techniques for detection of molecules in living cells: 1. Fluorescence microscopy and live cell imaging. Uses fluorescent dyes, genetically encoded fluorescent proteins, or labeled antibodies to detect specific biomolecules in live cells. Example: green fluorescent protein (GFP) tagging allows visualization of proteins in real time. 2. Fluorescence resonance energy transfer (FRET). Measures interactions between biomolecules using two fluorophores that transfer energy when they are close together. Used to study protein-protein interactions, enzyme activity, and receptor signaling. Example: FRET-based calcium sensors detect calcium ion concentration changes in neurons. 3. Bioluminescence imaging (BLI). Uses bioluminescent proteins like luciferase to detect molecules in live cells. Highly sensitive for tracking gene expression and cellular metabolism. Example: firefly luciferase reporters in cancer research to track tumor growth in live cells. 4. Fluorescent biosensors. Genetically engineered molecules that change fluorescence in response to a specific cellular condition, e.g., pH, ATP, Ca² levels. Example: genetically encoded calcium indicators, GECIs, like GCAM detect calcium flux in neurons. 5. Single molecule imaging, SMI. Uses super-resolution microscopy to visualize individual molecules in living cells. Example: tracking individual mRNA molecules using MS2-GFP tagging. 6. Fluorescence activated cell sorting, FACS, uses flow cytometry to detect and sort live cells based on fluorescent signals. Detect specific molecules on or inside cells using fluorophore labeled antibodies. Example: sorting immune cells based on CD markers. 7. Raman spectroscopy and surface-enhanced Raman spectroscopy serves label-free method to detect molecular fingerprints inside live cells. Detects proteins, lipids, and nucleic acids without requiring fluorescence. Example: monitoring drug accumulation in cancer cells. Applications of live cell molecular detection. Cellular signaling studies, detecting kinase activity, calcium waves, and neurotransmitter release. Cancer research, tracking oncogene expression, apoptosis, and drug response. Neuroscience, monitoring synaptic activity, ion flux, and neurodegenerative disease markers. Stem cell research, identifying pluripotency markers and differentiation pathways. Pathogen detection, identifying viral proteins, bacterial toxins, and immune responses in infected cells. Previously asked CSIRNet question CSIRNET 2022. Which technique allows real-time tracking of protein movement in live cells? A. Western blotting. B. Fluorescence recovery after photobleaching, FRAP. C. ELISA. D. Immunohistochemistry. Answer: B. Fluorescence recovery after photobleaching, FRAP. CSIRNET 2020. Which fluorescent protein is commonly used to study protein localization in live cells? A. Luciferase. B. GFP. C. DAPI. D. Ethidium bromide. Answer: B. GFP. CSIRNET 2018. Which technique is best suited to detect intracellular calcium levels in live cells? A. Mass spectrometry. B. FRET-based biosensors. C. SDSPAGE. D. Southern blotting. Answer: B. FRET-based biosensors. The ability to detect molecules inside living cells is essential for understanding dynamic cellular processes and developing new therapeutic strategies. You said. Circular dichroism. ChatGPT said. Circular dichroism. CD. Spectroscopy. Circular dichroism CD spectroscopy is a biophysical technique used to study the secondary structure, folding, and conformational changes of biomolecules, primarily proteins, nucleic acids, and chiral molecules. It is widely applied in structural biology, protein chemistry, and pharmaceutical research. Principle of circular dichroism. CD spectroscopy measures the difference in absorption of left and right circularly polarized light by chiral molecule. Chiral molecules interact differently with circularly polarized light, producing a CD spectrum. The shape and intensity of the CD spectrum provide information about the structural features of biomolecules. Applications of circular dichroism spectroscopy. 1. Protein secondary structure analysis. CD spectra can determine the proportion of alpha helices, beta sheets, and random coils in proteins. Example: alpha helix-rich proteins show strong negative bands at 208 nanometers and 222 nanometers, while beta sheets show a negative band at 218 nanometers. 2. Protein folding and stability studies. CD is used to monitor protein unfolding and refolding under different conditions like temperature, pH, or denaturance. Helps in studying the effects of mutations or ligand binding on protein conformation. 3. Nucleic acid structure analysis. CD spectra reveal A form, B form, or Z form DNA and detect RNA folding patterns. Example: bDNA exhibits a positive peak at 275 nanometers and a negative peak at 245 nanometers. 4. Ligand binding studies. CD can detect conformational changes upon drug or ligand binding to proteins or nucleic acids. Example: CD is used to study protein-ligand interactions in drug design. 5. Structural comparison of proteins. Used to compare the conformation of recombinant proteins with their native counterparts. Essential for biopharmaceutical quality control, e.g., biosimilar drugs. 6. Monitoring protein aggregation. Aggregation-related changes in secondary structure can be tracked using CD. Useful in studying neurodegenerative diseases like Alzheimer's and Parkinson's. Advantages of CD spectroscopy: non-destructive, does not alter the sample. Rapid and sensitive, provides structural information in minutes. Requires small sample volumes, ideal for studying biomolecules in solution. Can be used under physiological conditions, studies proteins in their native environment. Limitations of CD spectroscopy. Requires optically pure samples, contaminants can interfere with measurements. Limited resolution, does not provide atomic level details like X-ray crystallography or NMR. Signal intensity is weak, requires a high concentration of biomolecules for accurate detection. Previously asked CSIRNet question CSIRNET 2021. Which type of biomolecular structure can be analyzed using circular dichroism CD spectroscopy? A. Primary structure of proteins. B. Secondary structure of proteins. C. Tertiary structure of proteins. D. Quaternary structure of proteins. Answer: B. Secondary structure of proteins. CSIRNET 2019. Which of the following is a characteristic CD signal for alpha helical proteins? A. Positive peak at 275 nanometers. B. Negative peaks at 208 nanometers and 222 nanometers. C. Positive peak at 218 nanometers. D. No CD signal in the far UV region. Answer: B. Negative peaks at 208 nanometers and 222 nanometers. CSIRNET 2017. Which structural transition can be monitored using CD spectroscopy? A. Alpha helix to beta sheet conversion in protein speed DNA replication. C. Protein phosphorylation. D. Protein synthesis. Answer: A. Alpha helix to beta sheet conversion in proteins. Circular dichroism spectroscopy is a valuable tool for studying biomolecular structure, folding, and interactions, making it essential in both fundamental and applied research. You said. Fluorescence. ChatGPT said. Fluorescence and its applications in molecular biology. Fluorescence is a phenomenon where certain molecules, called fluorophores, absorb light at a specific wavelength and then emit light at a longer wavelength. This property is widely used in biological and biochemical research for detection, imaging, and quantification of biomolecules. Principle of fluorescence. A fluorophore absorbs light excitation at a specific wavelength. The excited molecule reaches a higher energy state. The molecule returns to its ground state, emitting light fluorescence at a longer wavelength than the absorbed light. The difference between the excitation and emission wavelengths is called the Stokes shift, which helps in detecting fluorescence signals. Fluorescence-based techniques. 1. Fluorescence microscopy. Uses fluorophore labeled molecules to visualize specific structures inside cells. Applications: protein localization, cellular dynamics, and live cell imaging. Example, GFP-tagged proteins allow real-time observation of protein movement in cells. 2. Immunofluorescence IF. Uses fluorophore conjugated antibodies to detect specific proteins in fixed cells or tissues. Types. Directive. Primary antibody is labeled with a fluorophore. Indirective. Uses an labeled primary antibody and a fluorophore labeled secondary antibody. Application. Diagnosing autoimmune diseases by detecting antibodies in patient samples. 3. Fluorescence-activated cell sorting FACS, a flow cytometry technique that uses fluorescence to analyze and sort cells. Applications. Identifying immune cells, detecting cancer markers, and isolating stem cells. 4. Fluorescence in situ hybridization. FISH uses fluorescent probes to detect specific DNA or RNA sequences in cells. Application. Identifying chromosomal abnormalities in genetic disorders and cancer diagnostics. 5. Forster resonance energy transfer. FRET measures molecular interactions by detecting energy transfer between two fluorophores. Application. Studying protein-protein interactions and enzyme activity. 6. Fluorescence recovery after photobleaching. FRAP analyzes protein mobility by bleaching a fluorescent signal in a region and tracking recovery. Application. Studying membrane protein dynamics. 7. Fluorescence polarization FP. Measures molecular interactions based on changes in fluorescence polarization. Application. Drug discovery and receptor ligand binding studies. 8. Single molecule fluorescence. Detects individual biomolecules, providing high-resolution insights into molecular behavior. Application. Studying transcription factor binding to DNA. Applications of fluorescence in biological research: cell imaging, detecting organelles, proteins, and nucleic acids. Molecular diagnostics. Identifying infectious diseases, e.g., tuberculosis using fluorescent probes. Cancer research. Studying tumor markers and metastasis. Neuroscience. Monitoring neurotransmitter release using calcium-sensitive fluorescent dyes. Genetics. Identifying mutations and chromosomal abnormalities. Previously asked CSIR Net question CSIR NET 2021. Which of the following techniques is based on fluorescence and is used to detect protein-protein interactions? A. Western blot. B. ELISA. C. PRET. D. PCR. Answer. C. PRET. CSIR NET 2019. Which fluorophore is commonly used for live cell imaging? A. Ethidium bromide. B. DAPI. C. GFP. D. Kumasi blue. Answer. C. GFP. CSIR NET 2017. Which technique uses fluorescently labeled probes to detect specific DNA sequences? A. Northern blot. B. Western blot. C. FISH. D. EMSA. Answer. C. FISH. Fluorescence-based techniques are essential tools in modern molecular biology, allowing researchers to visualize and quantify biomolecules with high sensitivity and specificity. You said, NMR. ChatGPT said. Nuclear magnetic resonance, NMR, spectroscopy. Nuclear magnetic resonance, NMR, spectroscopy is a powerful biophysical technique used to study the structure, dynamics, and interactions of biomolecules like proteins, nucleic acids, and small metabolites. It provides atomic level resolution and is widely used in structural biology, drug discovery, and metabolomics. Principle of NMR spectroscopy. Certain atomic nuclei, e.g., H, cube C, to the power of 5n, cube P possess nuclear spin, which aligns in an external magnetic field. When exposed to radio frequency RF pulses, these nuclei absorb energy and transition between spin states. The relaxation of nuclei back to their original state releases energy, which is detected and analyzed to generate an NMR spectrum. The chemical environment of each nucleus influences its chemical shift delta, which provides structural information. Types of NMR spectroscopy: 1. HNMR, proton NMR. Used for analyzing organic molecules, proteins, and metabolites. Provides information on chemical structure, hydrogen bonding, and functional groups. Chemical shifts range from 0 to 10 ppm for organic compounds. 2. Cube-C NMR, carbon-13 NMR, detects carbon atoms in biomolecules. Helps in determining molecular frameworks and distinguishing between different carbon environments. Typically uses proton decoupling to simplify spectra. 3. To the power of 5n and cube-P NMR. To the power of 5n NMR, used in protein structure analysis and nitrogen-containing biomolecules. Cube-P NMR, essential for studying nucleotides, ATP, phospholipids, and phosphorylation processes. 4. 2D NMR, two-dimensional NMR. Provides detailed structural information by correlating signals from different nuclei. Common techniques. Cozy correlation spectroscopy identifies spin coupling between protons. HSQC, heteronuclear single quantum coherence, correlates H with cube C or to the power of 5n. NOESY, nuclear Oberhauser effect spectroscopy, provides distance constraints for protein structure determination. 5. Solid state NMR. Used for studying membrane proteins, fibrils, and insoluble biomolecules. Requires specialized sample preparation and techniques like magic angle spinning MOS. Applications of NMR in biology: 1. Protein structure determination. Used to solve 3D structures of small to medium-sized proteins and LT, 30 kDa. Helps in understanding protein folding, stability, and conformational changes. 2. Drug discovery and binding studies identifies drug-protein interactions by monitoring chemical shift changes. Techniques like STD NMR, saturation transfer difference NMR, help in screening potential drug candidates. 3. Metabolomics and biomarker discovery. Used to analyze metabolic profiles in biological samples like blood, urine, and tissues. Helps in detecting disease biomarkers, e.g., cancer, diabetes. 4. RNA and DNA structure analysis. NMR reveals base pairing, secondary structures, and interactions in nucleic acids. Useful in studying RNA aptamers and riboswitches. 5. Enzyme mechanism studies. Tracks substrate binding, catalytic intermediates, and conformational changes in enzymes. Advantages of NMR spectroscopy: non-destructive. Samples remain intact for further studies. Provides atomic resolution data, essential for structural biology. Works in physiological conditions. Unlike X-ray crystallography, NMR can study molecules in solution. Detects molecular interactions in real time, ideal for drug discovery and biomolecular dynamics. Limitations of NMR spectroscopy: requires high sample concentration, typically in EM range. Expensive and time-consuming, requires high field magnets and specialized expertise. Limited to small to medium-sized proteins, larger complexes, and GT, 50 kDa are difficult to analyze. Previously asked CSIR Net question CSIR NET 2021. Which of the following nuclei is commonly used in biomolecular NMR spectroscopy? A squared C. B cube C C to the power of 6 OT cube squared P. Answer: B cube C. CSIR NET 2019. What is the role of NOESY in NMR spectroscopy? A. Identifies hydrogen bonding patterns in proteins. B. Provides distance constraints for 3D structure determination. C. Detects spin coupling in small molecules. D. Measures molecular weight of biomolecules. Answer: B. Provides distance constraints for 3D structure determination. CSIR NET 2017. Which type of NMR technique is used to study protein-ligand interactions in drug discovery? A. Cozy. B. STD NMR. C. MOS NMR. D. Department NMR. Answer: B. STD NMR. NMR spectroscopy is a crucial tool in molecular biology, providing detailed insights into biomolecular structure, function, and interactions. You said, ESR. ChatGPT said. Electron spin resonance, ESR spectroscopy. Electron spin resonance, ESR, also known as electron paramagnetic resonance, EPR, is a spectroscopic technique used to study unpaired electrons in biological and chemical systems. Unlike NMR, which detects nuclear spins, ESR detects electron spins and is useful in studying free radicals, transition metal complexes, and metalloproteins. Principle of ESR spectroscopy. Unpaired electrons possess intrinsic spin and behave like tiny magnets. In an external magnetic field, the unpaired electron can occupy either lower energy parallel or higher energy antiparallel state. When exposed to microwave radiation, the electron absorbs energy and transitions between spin states. The absorption signal is detected and plotted as an ESR spectrum, revealing information about the electronic environment. The resonance condition is given by H nu equals G beta B, where H equals Planck's constant, nu equals microwave frequency, G equals G factor depends on the electron's chemical environment, beta equals Bohr magneton, B equals magnetic field strength. Types of ESR spectroscopy. 1. Continuous wave ESR, CWESR, measures steady state absorption of microwave radiation. Used for free radicals and metal complexes in biological systems. 2. Pulsed ESR. Uses short microwave pulses to measure relaxation times in electron interactions. Provides higher resolution and structural information on protein dynamics and spin-labeled molecules. 3. Electron nuclear double resonance, ENDOR. Combines ESR and NMR to study hyperfine interactions between unpaired electrons and nearby nuclei. 4. Double electron resonance, DRP-LDOR, measures long-range distances between two unpaired electrons, useful in protein structural studies. Applications of ESR in biology. 1. Free radical detection. Identifies reactive oxygen species, ROS, and reactive nitrogen species, RNS, in biological systems. Useful in studying oxidative stress in diseases like cancer and neurodegeneration. 2. Metalloprotein analysis. ESR detects transition metal ions in proteins, e.g., Fe cubed, Q squared, Mn squared. Helps in understanding metal enzymes like cytochrome C oxidase and superoxide dismutase SOD. 3. Protein structure and folding. ESR with site-directed spin labeling, SDSL, provides structural insights into membrane proteins and conformational changes. 4. Drug-protein interactions. ESR studies how pharmaceutical compounds interact with metalloproteins and radical species. 5. Radiation biology. Measures DNA and protein damage caused by ionizing radiation and environmental pollutants. 6. Lipid membrane studies. ESR spin labeling tracks lipid mobility and membrane dynamics in biological membranes. Advantages of ESR spectroscopy. Highly sensitive to free radicals and transition metal complexes. Can study biological samples without extensive preparation. Provides structural and functional insights into proteins and enzymes. Limitations of ESR spectroscopy. Only detects paramagnetic species, substances with unpaired electrons. Requires low temperatures for high-resolution measurements. Less commonly used compared to NMR and X-ray crystallography for biomolecular studies. Previously asked CSIR-NET question CSIR-NET 2021. Which spectroscopy is most suitable for detecting free radicals in biological systems? A. NMR. B. ESR. C. UV. B. Mass spectrometry. Answer. B. ESR. CSIR-NET 2019. What is the principle behind ESR spectroscopy? A. Absorption of microwave radiation by unpaired electrons in a magnetic field. B. Absorption of radio waves by atomic nuclei. C. Absorption of visible light by conjugated molecules. D. Emission of X-rays from heavy metals. Answer. A. Absorption of microwave radiation by unpaired electrons in a magnetic field. CSIR-NET 2017. Which biomolecule can be studied using ESR spectroscopy? A. DNA double helix. B. Free radical intermediates in enzymatic reactions. C. Ribosomal RNA. D. Lipopolysaccharides. Answer. B. Free radical intermediates in enzymatic reactions. ESR spectroscopy is a crucial tool in biophysics, structural biology, and free radical research, offering unique insights into electron-rich biomolecules and paramagnetic species. You said. Molecular structure determination using X-ray. ChatGPT said. Molecular structure determination using X-ray crystallography. X-ray crystallography is a powerful technique used to determine the three-dimensional structure of biomolecules such as proteins, nucleic acids, and small organic compounds. This method provides atomic-level resolution, making it essential for drug discovery, structural biology, and material sciences. Principle of X-ray crystallography. When a crystal is exposed to a beam of X-rays, the atoms in the crystal cause the X-rays to scatter. The scattered X-rays interfere constructively and destructively, producing a diffraction pattern. Using Bragg's law, the diffraction data is analyzed to reconstruct the electron density map, which reveals the molecular structure. Bragg's law, n lambda equals 2 d sin theta where n equals integer order of diffraction. Lambda equals X-ray wavelength. D equals spacing between atomic planes in the crystal. Theta equals angle of incidence. Steps in X-ray crystallography. 1. Crystal growth. The molecule of interest must be purified and crystallized. Crystallization methods include vapor diffusion, microbatch, and seeding techniques. 2. X-ray diffraction. The crystal is exposed to a monochromatic X-ray beam. The diffraction pattern is recorded using a detector. 3. Data processing. The diffraction data is analyzed mathematically using Fourier transform to generate an electron density map. 4. Model building and refinement of a molecular model is constructed to fit the electron density. The structure is refined to improve accuracy. Applications of X-ray crystallography. 1. Protein structure determination. Provides atomic resolution structures of proteins, enzymes, and receptors. Helps in understanding enzyme mechanisms, protein folding, and conformational changes. 2. Drug discovery. Determines how small molecules bind to proteins, aiding in rational drug design. Used to develop inhibitors for HIV protease, cancer targets, and bacterial enzymes. 3. DNA and RNA structure analysis. Led to the discovery of the double helix structure of DNA, Watson and Crick, 1953. Helps in studying RNA folding and ribosome structure. 4. Material science and nanotechnology used to study the structure of minerals, catalysts, and nanomaterials. Advantages of X-ray crystallography provides high-resolution atomic details. Works for large biomolecules that cannot be studied by NMR. No size limitation, unlike NMR spectroscopy. Limitations of X-ray crystallography requires high-quality crystals, which can be difficult to obtain. Cannot S.